And so for part 3 of this lecture, we will talk specifically about how this idea of heritability can translate into behavior evolving. And so we'll start by looking at a few animal examples, and then we'll talk about humans. And so to address whether behavior can evolve, we're going to start by looking at dungfly. These were studied by Jeffrey Parker. Dungfly females mature a large quantity of eggs and then store those eggs until they're ready to oviposit or lay those eggs on a pile of dung. And so typically they can deposit 100 or upwards of almost 1,000 eggs in one kind of period of oviposition. They look for a suitable dung to place their larvae in. They first fly to and fly past on the upwind side a newly deposited dung. Then they cautiously move to the dung surface to oviposit. They are also able to store sperm, but despite this, they have no problem finding many males. So therefore, the cowpat and more specifically, the female reproductive tract are a site for sperm competition. Males will flock to newly laid dung. 4 to 5 males will be present on a pile of dung for every female who's present. The male will sight the female, leap, mount, orient themselves, and then mate immediately. Dungfly have a particular penis morphology that is like a plunger to be able to force out old sperm. Success in terms of the proportion of eggs of that female that are fertilized by a particular male depends on how long males copulate. After ejaculation, males will hang on to the female while she's ovipositing. They will also then have to competitively defend that female. Other males will try to attack the mated pair and displace the male who just copulated. The male will guard and fight off other males with their middle legs while holding on to the female with their first pair and third pair of legs. What then is a male to do about all those other males? So we're going to look specifically at the decisions that males make and then at how they solve those decisions. There are 3 types of females from the male point of view. There are those that are just arriving at the dung, those who are already copulating but have yet to oviposit their eggs, and those who are currently ovipositing. Arriving females are the best bet because they're free for copulation. They have not yet been claimed by a particular male. Copulating females are your second best bet. There will be a struggle as they have a male defending them, but they haven't oviposited yet. Ovipositing females are your least successful bet because they only have a few eggs that are left to be fertilized. Most of the eggs have already been fertilized. And so in the first 20 minutes surrounding a new dung being laid, females are in 5 zones. They are on the dung surface, in the grass area surrounding the dung within 20 centimeters, which is about 8 inches, between 20 and 40 centimeters from the edge of the dung, between 40 and 60 centimeters, and from between 60 and 80 centimeters. So we will look at where males position themselves first, then we will look at the duration of copulation. Immediately following the deposition of dung, where would you expect the males to be? Well, females who are already on the dung surface will be ovipositing. So the arriving females are going to be concentrated in this B zone, this period or this space from about 0 to 20 centimeters surrounding the dung. The farther away that females are, the less competition males may face from other males. But at some point, when you move pretty far away, there's not really much benefit of even trying to mate with a female because you're not certain that they are ready to oviposit. So in that that period immediately after a new pile of dung is deposited, you find most of the males in Zone B, this zone of arriving females. Those males who are in Zone B have the greatest likelihood of capturing a female and then copulating with her. The second greatest likelihood of capturing a female and copulating is with females who are on the dung surface, and then your success goes down as you move farther and farther from the dung. And so we are able to mathematically model male choices, and then we are able to observe males and track what they actually do so that we can compare our observed and expected values. And as you can see from this graph, they line up pretty well. Males are doing exactly what our mathematical models predict that males would do. That is, that they cluster in zone B, then zone A, and fewer males with less success in C, D, and E. Similarly, we know that the likelihood of fertilization depends upon how long males spend in copulation with females. And so we will specifically talk about this kind of model later on, but this model reflects what's called the marginal value theorem. That is that the payoff of a male's decision, or the payoff of anyone's decision, an individual's decision, is based on not only the time that they may spend dedicated to a particular activity, that could be foraging, that could be copulation, but also the amount of time that it would take them to search for a new opportunity, a new situation where they can exhibit that behavior. And so the marginal value theorem has been used, for example, looking at starlings with how many prey they load in their mouths based on how far apart feeders are. It's been used for humans for foraging decisions, specifically regarding what we call patch choice, or how long individuals should stay in a particular patch. And so here is another example of the marginal value theorem. That is the amount of time in this specific example that males should spend in copulation, given search time to find another female with whom to copulate. And so again, we can mathematically model what we would expect that decision to look like, and we can predict an optimal copulation time of roughly 40 minutes, and then we can track what dung flies do, what they actually do, and we find a copulation time, an observed copulation time of about 35.5 minutes. So dung flies are making both positioning decisions and copulation decisions based on the relative payoffs of those particular decisions. Now, the thing that you want to keep in mind here when we talk about whether behavior can evolve is that these dung flies, in this context, they're not hardwired for this behavior. So, in some cases, and in the next case with cricket calling times, we'll discuss this particular example, but in some cases, the decisions that an organism makes, an individual makes, are dependent specifically, well, it's not really a decision, let's say, so that there may be some behaviors, and we'll talk about specifically the cricket calling times, that are genetically based, and so they possess a gene for a certain calling time, and they don't really deviate from that calling time. That is going to be a vast minority of cases. Typically, when we talk about behavior evolving, we're talking about conscious decision making. And so here with dung flies, they're not genetically coded to always wait in zone B, or to always wait in zone A, or to always wait farther from the dung. So they are making individual-based decisions specifically that are context-specific, that are cued in or based upon what the conditions are at that particular moment. And so the very next time that they have an opportunity for copulation, the decision they make might be totally different. And this is much more an appropriate way to model behavioral decisions, because this is also what we would expect to hold true with humans, right? We don't expect that humans are going to be specifically hardwired for a certain behavior. We expect rather that humans will have quite a bit of what we call behavioral flexibility. And so we're going to keep talking about that as we go through these examples. Dung flies certainly demonstrate behavioral flexibility in that they will alter their copulation time, they will alter the zones in which they wait based on these specific contexts, or number of competing males, or number of females who are present, or search time necessary to gain new new females with whom to copulate each and every time they go to mate. So we can also show that natural selection operates on behavior by looking at some genetically based behaviors. And so William Cade looked at cricket calling times, and he took males that had different calling times. They were specifically categorized into long callers and short callers, and then he separated them out into the two most extreme ends of the distribution. And so this would be, if this were natural selection that we're selecting for calling times, this would be the disruptive selection, where that mean calling time is disfavored, and it's the two extremes that are favored. So he developed two populations of crickets, long callers and short callers. He then mated each of those populations with females at random. And after one generation, there was already a statistically significantly different value in the calling times of those offspring. So really already with only one generation of mating, we see a distinction between, at the population level, long callers and short callers. Typically we would expect a bit more time necessary for us to see two discrete populations. Are there any examples of environmentally contextual situations where calling time may vary, and specifically vary in a way that would select for either calling times or short calling times? Well, there are. There's a particular kind of fly that parasitizes crickets. And so with this particular kind of context where flies may be parasitized and thusly then die and their babies be eaten. When flies are absent, we see natural selection favor long calling times. When flies are present, we see natural selection favoring either no calling or short calling times. So the presence or absence of flies in this context is the mechanism of natural selection. And then, of course, the outcome is whether those populations then are long callers or short callers. So we can specifically pinpoint a naturally occurring context during which we might see natural selection favor one end of the distribution or the other. Do we see a genetic basis for some human behavioral tendencies? Well, the answer is absolutely yes. And so here we're specifically looking at the heritability of mental health issues, of neurologically based kinds of challenges that people may face. And when we want to determine human heritability, the best way to kind of calculate the degree to which certain behavioral tendencies are heritable is to look at twin studies. And so ideally we would have identical twins that we reared in different environments, and we'd measure kind of the output or the similarities and differences from those two different environments. But the population, the potential population of what we call monozygotic twins, that is identical twins, we've got small population numbers. There simply aren't that many identical twins conceived. And then particularly to stratify them to different rearing experiences. I mean, most families who have identical twins raise them in the same environment. So we don't have that particular kind of context with which we can compare. So the other way to get at heritability values is to look at monozygotic or identical twins versus dizygotic or fraternal twins. And so comparing across these two populations, presumably dizygotic twins will be different in terms of genome, so their genotype will vary, but their environment would be consistent. So this gets at even the potential differences that we might see in the intrauterine environment. This will control for that, because the intrauterine experiences that twins have should be fairly similar because they're gestating at the same time. So we look for then a difference between the concordance among dizygotic twin pairs and the concordance among monozygotic twin pairs. And when we see significantly greater concordance between identical twins than we do fraternal twins, we can pinpoint a root genetic cause because, of course, identical twins are similar, or well, I mean, by definition, identical in their genetic composition, whereas fraternal twins differ with their genotype, but are reared in that same environment. And so, for a lot of psychiatric conditions, for a lot of mental health issues, we find a very high degree of heritability. So, this includes borderline personality disorder, mania, manic depression, schizophrenia, a mood disorder spectrum, so on and so forth. There certainly are genetic tendencies towards even things like depression and anxiety. Now, of course, those can also be quite plastic and context specific. A death in the family might push some people towards acute depressive phase, whereas other people maybe have more resilience based on rearing experiences as children, etc. So we're not asserting that every aspect of mental health is based on a heritable component, but rather that there is strong heritability of certain kinds of mental health challenges. And so if we can pinpoint that heritability affects someone's personality or mental health, then we can make that next leap to state that, or hypothesize that, heritability would also affect individual kind of behaviors. Now, does that mean all behavior is genetically determined? Of course not. We are incredibly flexible, incredibly plastic in our behavior. We'll talk about that in a few moments. So we're going to look at one more animal example with bluegill sunfish to address how predictable environmentally flexible behavior can be, because we've already determined that that dung flies are variable or flexible in their where they wait for females to mate and how long they spend in copulation. So we have this context already where we see environmental flexibility. So the question then becomes, can we predict, as we mathematically model things, can we predict the different decisions and patterns of decisions and patterns of behavior that flexible creatures may make? Because if we can predict it, then we probably have a pretty good understanding of how environment triggers particular behaviors. And so with bluegill sunfish, there are two phenotypes for adult males. These are males that have the ability to produce gametes. This is how we're measuring adulthood. It's not a sheer body size kind of relationship. And so males can either be parentals, or in this picture, large male, or they can be cuckolders, or in this picture, satellite males. And so the payoff to the cuckolding phenotype depends quite a bit on the particular environmental features. So we can predict the number of cuckolders that would be present given certain environments. We can also predict an age-related response such that younger males are more likely to demonstrate this cuckolding phenotype than older males. And so what do we see? Well, this cuckolding phenotype, the satellite males, it's a case of female mimicry. And so you see that the parental male, the large male, is a different color, and we also see body size sexual dimorphism. They're much, much larger than the females are. The satellite male is indistinguishable from females. So it's not going to be perceived in the same way that females may perceive these large males. He's not going to be perceived the same way by the females. They may let these males get closer to them, even particularly during periods where they might not be interested in copulation. Also, he's going to elicit a different behavioral response from that parental male as well, from that large male. Large males, when they are encountered with other large males, they fight, and they fight quite violently. When they are encountered with males that look female, they basically ignore them. It's a case where that female mimicry confers a level of protection. And so with sunfish, we find that the success of cuckolders depends upon the percent cover, such that when there is more cover, there will be fewer cuckolders. When there is less cover, it pays off to be a cuckolder. And so what's driving this relationship? Well, when it's a weedy kind of pond, when we have a lot of places to hide, the benefit to the individual male is such that he's going to have greater success if he presents this large male phenotype. And really that's because he, as a large male, can hide, right? When there's a large degree of cover, he's going to be successful at not eliciting a violent response from the resident male because the resident male is not going to be able to see him. When that percent cover is a lot less, so it's a pretty open area in the lake, then we see the payoff to cuckolders increase dramatically. And so when there's no place for males to hide, they're not going to be successful. If they try to intrude upon a male's territory with this full adult body size, they're going to end up in very violent contests over who kind of wins out and who has those females mate with them. So in that kind of context where specifically there's a chance that those males will be closely observed by the resident parental male, the benefit or the higher benefit in terms of mating success comes from this cuckolding phenotype. So lots of cover, few cuckolders. Very little cover, many cuckolders. When we look at the life histories of male bluegill sunfish, we also find that there tends to be what we call an ontogenetic rule. That means a decision that's made not based on specific environmental circumstances like this percent cover, but rather based on age and stage of development. And so fish being cold-blooded have potentially much slower growth rates. And so the rule of thumb ontogenetically here is that if you are very young, specifically yearling to about 6 years of age, then reduce the likelihood you're going to get beaten up by other males by adopting this cuckolder phenotype. If you've survived already for at least 6 years and you're between your 6th and 7th year, then you've got this reasonable assumption of survival and go ahead and become a parental male. So the rule of thumb is when young, be a cuckolder, conserve that energy that you would otherwise devote towards large body size and fighting. Once you've lived past this point, make the decision to go ahead and change your phenotype. So 2 behavioral rules. One that deals with particular ecological environments and one that deals with ontogeny or stage of development. So how then do we translate genotype into phenotype? Remember that the genotype is the specific combination of genes possessed by an individual. It is your genetic code. It's a sequence of base pairs that codes for codons, that then code for amino acids, that then code for proteins. Phenotype is the actual suite of behaviors or characteristics or traits exhibited by living organisms. So your phenotype can vary predictably with genotype, and this is certainly in a behavioral context what we see with cricket calling times and the high heritability of these personality characteristics. So with respect to morphological features, it could be ABO blood type. That is a phenotype that varies predictably with your genotype. Could be eye color, would be less likely to be something like height. In other cases, though, phenotypes vary according to the environment, even when genotype is held constant. This is what we see with individual stature and overall body shape, right? That you don't, over the course of your lifespan, change your genotype, but you do grow taller. And you may have flexible body weight as well and have some context-specific times in your life where you're heavier than average and context-specific times in your life where you're lighter than average. We also discussed these examples of the waiting times and the zones for waiting among dungfly. And so here we've got this, the dungfly aren't varying their genotype from mating time to mating time from female to female, but they are making very different decisions about where to wait. We also see this with percent cover in sunfish, that payoff. If we move a sunfish, a small male sunfish, from a context, an environment that has a lot of cover to an environment that has very little cover, we might arrest his overall growth rate so that he stays as mimicking females, as this cuckolder male. If we move him from a situation where there's a lot of cover to one that has, or a situation where there's very little cover to one that has a lot of weedy cover, then we might trigger a rapid period of development where he grows in overall body size and changes his coloration very rapidly with these changing ecological characteristics. So we're not changing the genotype, we're simply changing the environment that that genotype is expressed in. And age is another factor. The sunfish genotype doesn't change from age 6 to age 7. Rather, the period of time that you've been alive does, and that triggers then this divergence or this maturation into parental males from cuckolder males. So what this does is gives us the concept of what we call reaction norms. That it's not a particular phenotype or the behavior itself that is selected for. Rather, what is selected for is the ability to change your behavior given different environmental and personal characteristics. And so environment here with reaction norms comes in at 2 points. It helps determine the nature of how that genotype translates into phenotype. So, that percent cover situation, that the genotype of a male is going to either be parental or cuckolder based on what that, or the phenotype of the male will be parental or cuckolder based on how much cover there is in the particular area of the pond where, or lake where the sunfish finds itself. But then environment also selects among phenotypes. So there's going to be some sort of environmental selection pressure through which natural selection operates. And so, for example, body size. Nutrition is a way in which phenotypes can be determined. So if during critical periods of development you don't have access to enough food, enough calories, you're not going to develop to your full genetic potential. You're not going to be super tall and really fit and buff, right? If you have exposure to way too many calories, particularly calories that are processed that are relatively unhealthy, then you might grow to your potential, full potential in stature, but you might put on excess body weight as those excess calories are translated into fat. So nutrition is one good example of how the environment can select among different phenotypes. So when we talk about evolved reaction norms and when we talk about phenotypic plasticity, we are talking about the same thing. It is not an invariant expression of a behavior that is selected for. It is the range of behaviors given different environmental circumstances. So it's the reaction pattern to the environment that evolved rather than a single invariant phenotype. The pattern of phenotypes associated with different environments is called the reaction norm. And the ability to modulate your phenotype based on different environmental input is called phenotypic plasticity or facultative phenotype. So the pattern of behaviors is your reaction norm. The ability to change your behavior is a facultative phenotype or phenotypic plasticity. Reaction norms are expected to be adaptive because they evolve by natural selection. So it is not that natural selection shapes one particular pattern of behavior, but rather that natural selection selects for a suite of behavioral expressions. And so we'll look at just a few examples before we finish part 3 of this lecture. This is an example of a reaction norm to temperature for 3 different genotypes of Drosophila or fruit flies. Wild type Drosophila are those that you find in nature. These infra and ultra bar Drosophila are lab variants. And so among wild type Drosophila, here we're looking at the number of facets in the eye. So remember that insects have multifaceted eyes, unlike humans. If you've ever gone to the Natural History Museum here in Albuquerque, they have that little like praying mantis or fly eyes that you can look through and you get fractured images because of all those facets. So insects see quite differently than we do. The typical reaction among wild type Drosophila is such that at higher temperatures there are fewer facets on the eye. We see a similar relationship for ultrabar Drosophila, that higher temperature leads to fewer facets, but notice the scope of that. At 30 degrees Celsius, wild type Drosophila will have 750 to 800 facets. Ultrabar Drosophila will only have about 60 facets, so that scope is different. Then contrast this to these infrabar lab-selected Drosophila. When they see increasing temperature, they increase the number of facets. So at a temperature of about 15 degrees to about 17 degrees Celsius, infrabar and ultrabar will have very similar eyes, but at temperatures of 25, 30 degrees Celsius, infrabar will have more facets, ultrabar will have very few facets. We see similar kinds of reaction norms with one trait that humans possess called red blood cell density. So red blood cell density is an adaptation to oxygen-poor environments that typically come at high elevations. So, of course, as you go from sea level up to a 10,000-foot mountain, you're going to find it harder to breathe up at 10,000 feet. Or if you're going to summit Everest, most Westerners who summit Everest have to do so with supplemental oxygen. Now, you will establish base camps and kind of acclimatize yourself to those conditions, but we simply don't have a genetic basis that allows us to breathe in really oxygen-poor environments like 30,000 feet. So there are multiple ways that we can adapt to this low oxygen concentration. One is genetic. If you look at the Nepalese Sherpas who help with summiting Everest, I mean, many of them can summit without supplemental oxygen because they've lived at those high elevations for a very long period of time. Similarly, we can use supplemental oxygen, and we do that all the time when we fly in airplanes. The cabin will be pressurized, and in case of a loss of cabin pressure, oxygen masks will drop from overhead. But we have a very kind of short-term physiological adaptation here that is the ability to change our red blood cell density. So red blood cell density, in a unit volume of blood, is going to vary based on whether you find yourself at low elevation or high elevation. This is one of the reasons why many endurance athletes pick places like Albuquerque, Denver, Boulder for their training, because after about 2 or 3 weeks of exercising at high elevation, you've basically done legalized blood doping. You've basically increased your number of red blood cells per unit volume blood. And what that does is gives more hemoglobin for scant oxygen particles to bind to, so that every bit of oxygen that is present in a unit volume air that you take in will be bound with hemoglobin and be able to be used by your cells. We see this depicted in these 2 girls, one on the left, a girl from the Alps in Switzerland, and on the right, a girl from the Andes in Peru. If you notice, both of them have very red cheeks. It's not because they've just been running. It's also not because they're incredibly cold. It is because their unit volume of blood has more red blood cells, so it makes their cheeks take this pink hue. And so natural selection does not select for a particular invariant phenotype. Rather, it selects for a phenotype generating mechanism. In a low oxygen environment, the output is going to be high red blood cell density. In a high oxygen environment, the output is going to be low red blood cell density. So it is this pattern of phenotypes that is selected for this ability to modulate one's phenotype given different environmental input.